Right.
Jonathan's one of rising stars, and apart from his pediatric neuroimaging work, he's the co-director of a newly established Center for Intergenerational Psychiatry with Christine
Cool.
Duarte.
Nice.
So right up your alley.
Yeah, that's cool.
Okay, well, thank you. We're going to get started. Good morning, everybody, and welcome to Grand Rounds. I believe you're looking at my screen, which has a photo in front of it. Simon, if that's the case, say yes.
Yes, it is.
That's
Okay.
correct.
Good, good. So I'm going to forego the usual announcements simply because of the fact that our interface with the virtual WebEx platform we're using is a little clunky, and I don't want to get things derailed. Other than to say that next week our speaker will be Craig Spencer, who will be talking about the experience in family medicine and the emergency services of COVID, and then following the week after, our own Catherine Elkington. But this week we have the privilege and good fortune to have a very busy man, Dr. Ned Kalin, who resides in Wisconsin, which has been the center of a lot of activity lately and also historically. And I believe the population, the student body population of the university is what, like 40,000?
A little over that.
Yeah, so it's big, and it's really a barometer for a lot of the social trends in the United States, in addition to being a site of some of the elite academic scholars in the country. But Ned hails from the East Coast and then moved west. He went to undergraduate school at Penn State, his medical school in Philadelphia at Jefferson. He then did his training at the University of Wisconsin, which included a stint at the NIMH and as a research fellow, where he learned using, for the purposes of neurobiological studies, animal models, including subhuman primates. And then he returned to the University of Wisconsin, where he ascended to his current role, which is the Hedberg Professor and Chair of Psychiatry at the University of Wisconsin, and also a slew of other titles that he holds within the university as the director of various programs and centers, in addition to his administrative responsibilities. Ned has been one of the sort of generation of biological researchers in psychiatry, sort of to probe the brain and its neural systems to try and determine how emotional regulation, temperament develop maturationally, and how those are affected by genes, constitution, or experience in ways that produce vulnerability or resilience to various psychiatric disorders, and particularly in the mood anxiety realm. And the key element of it is that by being able to work across species translationally, he's been able to do it in a way that maps the trajectory over the life cycle, which is really critical given the origins of most psychiatric disorders as a beginning in childhood or in early life. It's also very relevant to the approach that's been newly supported within our own department and institute that Christine Duarte and Jonathan Posner are leading, which is the Center for Intergenerational Psychiatry. Ned's work is reflected in numerous publications. His first publication actually was a publication in one of the medical journals on the frustration of a newly minted psychiatrist. We can all relate to that. And his last publication, though, was in PNAS on the, I don't have the citation in front of me, but it was on amygdalar development in early life development exposed in individuals exposed to stress. So this shows you the scope of his work over his career. As a result of that, he's gained considerable distinction, including having been elected to the National Academy of Medicine. And also, as of, what was it, 2017, that you were appointed executive editor of the American Journal of Psychiatry?
2019.
2019. If you weren't friendly with Ned before, you want to be friendly with him now because he wields considerable editorial influence. But more important than any of that is the picture you're seeing here. Forget about the fact that it's a bunch of white men. It's some academic psychiatrists who are deluding themselves that they're real outdoors persons and can able to catch a fish. Of course, it's catch and release in this case. So Ned is in the middle there. He's the bespectacled individual smiling. This was a group of individuals who are known to probably many of you on the left. Far left is Alan Schatzberg, Charlie Nemiroff, next to him, Marty Keller, then Ned Kalin, then Clint Kiltz, and I'm not sure who that is on the right, but he was someone they picked up along the way. But this was a way that we were able to recreate and decompress from the pressures of academic medicine and have survived to tell about it. So it's a real pleasure to have Ned with us today and to present some of his more recent work. And I'm going to, with that, ask Simon to assist me in giving up my screen so that Ned can share his and show it. So Ned, the floor is yours.
Thank you very much, Jeff. Can you guys hear me? Jonathan, nod if you can. Okay, thank you. And I'm going to go to full screen, I think. I need to do that. So I can't see anybody now, but I'm assuming everybody can hear me and that you can see my slides. If Simon, for sure, if anything goes wrong, let me know, okay?
You just need to share your screen.
Everything's good?
No, you just need to share your screen.
Oh, that would be useful. Okay, let me figure that out. So I need to go back to, let's see, maybe.
Yep, hover your mouse. Oh, you got it.
I don't think I do have it. I'm going to end show.
Hover your mouse over the middle.
Yeah.
Box with the up arrow.
Yep, got that. And I'm looking for
PowerPoint.
PowerPoint.
There you go.
That come up?
Coming up. Yeah, there you go.
Okay, I'll go to full screen now. Is that working?
We can see it. Go to full screen, though.
I think I did go to full screen. What are you seeing? Are you seeing the other side?
Now, there you go.
Now it's good. Now we can see just your slides. Perfect.
Okay, thank you. All right, well, listen, it's a pleasure to be able to do this. Jeffrey, thank you for that really nice introduction, and also Jonathan, thank you for the invitation. This is it's obviously a strange format, and I talk about anxiety and anxiety disorders, and I'm telling you right now that I'm feeling a little anxious myself because I can't see any faces. So I'm going to try to imagine your faces all smiling at me and reinforcing me as I give this talk. And Simon, please let me know also if things aren't coming through right, okay? So what I'd like to do today is to give you an overview of what's been exciting to me over the years in relation to our field and how I've directed sort of my research program in relation to this. And this really is around trying to understand as best we can what the early biological and psychosocial factors are that come together to either increase risk or to increase vulnerability or increase resilience in relation to the likelihood of developing stress-related psychopathology, particularly anxiety and depression. And the work that I've been doing, as Jeffrey mentioned, has really been very translational, working across non-human primates, rhesus monkeys as an animal model, and also working with children and pre-adolescents to try to translate back and forth across species in ways that can be most helpful for developing a better understanding of the mechanisms and pathophysiology that underlie the early life risk to develop anxiety and depression, as well as with the hope of gaining insights that can result in new ideas about treatment strategies. Ultimately, that's what we're all about in our field. And I think the exciting opportunity from a developmental perspective is the concept that we can move our treatments earlier and earlier in relation to understanding the illnesses that we treat, with the sort of the ultimate goal of coming up with early intervention strategies that may mitigate the lifetime course that of suffering that many of our patients suffer from. These are my disclosures. I won't go over them now. Jeff mentioned that I'm the editor of the American Journal. I receive a stipend from them, and also have been involved with some advisory boards and a bit of consulting. None of this is directly related to what I'll be talking about today. So just as a review, I think what I'm going to do in my talk is I'm first going to talk a little bit about anxiety disorders, their prevalence, and why they're obviously important. I'm then going to move to talk about why development is important and why childhood is important from the standpoint of understanding anxiety disorders and stress-related disorders. Then I'm going to move to studies that we've done in non-human primates as well as children, showing hopefully illustrating for you the value of cross-species work from the standpoint of reverse translation and translation, learning more about mechanisms. And then finally, we'll end up with some more recent work that has allowed us to manipulate in the primate brain either circuits or molecules as a way to test new ideas about new potential treatment targets or even new potential treatments. Anxiety disorders, in a way, have been sort of the stepchild of major psychiatric disorders. There's obviously great attention to play to schizophrenia, to major depression, to bipolar disorder. Anxiety disorders, in some ways, have been relatively neglected, even though they're among the most common psychiatric disorders. All of you know that are clinicians that anxiety disorders come in different flavors from the standpoint of both types of disorder as well as severity. But when severe, can be very disabling. As I mentioned already, what we're up to is really trying to understand more about novel treatments based on mechanisms and pathophysiology and broadening our understanding of that. Now, I'm not a child psychiatrist, but my work has led me to this very developmental, child-oriented position, mostly because I believe that most of what we deal with in psychiatry has its origins very early in life, or at least many of its determinants, also, as we know, in utero. And that if we are wise, in addition to helping children with their current suffering, the hope here is that we can develop ideas about early detection and early intervention across the board in psychiatry that will be useful. In children, anxiety disorders are incredibly common. As I'll show you in a minute, they actually onset early in life. Most people that have anxiety disorders in adulthood, if you probe long enough or deep enough, you'll find that they've had significant anxiety as a child. Already mentioned they can be quite disabling, but also what's important to keep in mind is that childhood anxiety, when it is pathological, and I use that term broadly because I'm not just talking about DSM, meeting DSM criteria, I'm also talking about individuals that have persistent anxiety that is disabling but don't meet criteria. This actually can be a risk factor for other psychopathology, including other anxiety disorders, depression, and other stress-related psychopathology. This is just data from the NIMH website from Harvard Medical School, the National Comorbidity Survey, demonstrating that lifetime prevalence of anxiety disorders among adolescents is incredibly high, around 30%. In adulthood, the past year prevalence of anxiety disorders is estimated in this particular survey to have been around 20%. So very, very common. Now, one of the important features about anxiety and anxiety disorders is that it is virtually expressed and experienced with every psychiatric illness that we deal with. And we know that across the board, comorbid anxiety symptoms tend to make outcomes more difficult and worse for patients, regardless of the diagnosis. This is just demonstrating the high levels of comorbidity, lifetime comorbidity, that is seen with depression, with anxiety disorders in relation to depression. And so you can see these comorbidities are incredibly high over the lifetime, not a surprise to clinicians that deal with patients with anxiety or with depression. If you look at this from the other perspective, and if you take the major depressive disorder and ask how comorbid is anxiety disorders with major depression, you see basically the same kind of findings, around 36 or 40% lifetime prevalence of comorbid anxiety disorders with major depressive disorders. So these disorders are not necessarily independent of each other. Many people think that they're quite related from the standpoint of their pathophysiology and their treatment. They're clearly distinct as well, but highly commingled, if you will. If you look at the STAR-D study, level 1, that was the citalopram phase of the study, one of the findings that came out of that was that 53% of the patients had what was termed anxious depression. And importantly, out of that, those patients were less likely to respond as well to treatment. So this is just making the point that I already made, that comorbid or coexisting anxiety complicates treatment and can make treatment outcomes more difficult. So the point that I want to make and to emphasize is that anxiety disorders are common. They can be disabling. Anxiety symptoms and anxiety disorders are highly comorbid with other psychiatric illnesses. And in general, as I said, when present, tend to make treatment more difficult. Now, what I'm going to do now is actually show you a video of one of the young girls that was involved with one of our studies. We're studying pre-adolescent girls who either have a risk for developing anxiety or who have anxiety disorders. And this young girl was nice enough to allow us to tape her and to use this. And what I want you to listen to is her story a bit, but also pay attention to the comorbid symptoms that she talks about, just to give you a flavor for what this can be like in childhood. And I'm going to try to get this to work now. Let me just see here if I can move to this. Can you guys still see the screen? Simon, is that still there?
Yep. Yeah, we can see it.
Okay. Let me see if I'm going to go to internal speakers. And let's see if this will work now. So
mainly when I was
can you hear that okay?
kindergarten.
Yeah.
Yes? Okay, good.
Now the volume's too low.
And I used to have terrible, like fits of anxiety where I'd hide under my bed and say,
You're not hearing the volume, Ned.
I hate everybody.
More volume?
So terrible to me.
Yeah.
I think the part was thinking that everyone didn't like me. I would
Is that
always have
better?
this feeling of loneliness.
Not much.
I would really misread people, and I was sure that I was right when I was totally wrong. It was really scary and really sad, just the fact.
Put your headphone back down and see if it works.
Fantasize about why can't I have this life, and I feel like in my life,
Still not able to hear the volume very well.
were different than mine, but that made it for me seem better than it actually was. And so I think that it was just really
That's a little better. Why don't you start it over?
goodness my parents recognized it. Otherwise I wouldn't be here in front of cameras.
Yeah, I mean, we were actually worried about, things like suicide in kindergarten and first grade. It was terrifying. Yeah, and we're not really worried about it. Because you say, I don't want to exist, I wish I didn't exist. And we have a lot of kids, so we're not really alarmist. Not, a bomb has to go off to really phase us. But it was terrifying. Yeah.
I wouldn't want to hurt myself or die because both of those things scared
Yeah,
me.
I don't think it's
But I
coming
didn't
through.
want to just not exist, like poof into magical fairy dust air and never have to face anything again and be happy.
I don't think it's coming through. Simon, do you know what the problem is?
It was working. Go ahead.
Ned, the sound didn't come through audibly.
Simon, did you?
So if you could play it again and just turn the volume up on your computer all the way, and then just try it again.
I think it's up all the way now.
Okay, just push play then real quick.
All right, let me get back.
Mainly when I was younger, in kindergarten and first grade.
Can you hear that?
Yep.
Yeah, it's better.
All right, should I play it again? I'll play it again.
Yep.
Okay, great.
Mainly when I was younger, in kindergarten and first grade, it became very apparent that I had ADHD and anxiety, and so my parents started taking me to psychologists. And I used to have terrible, like fits of anxiety where I'd hide under my bed and say, I want to kill myself, I happened to me. I think that the hardest part was thinking that everyone didn't like me. I would always have this feeling of loneliness and abandonment, even if I would really misread people, and I was sure that I was right when I was totally wrong. It was really scary and really sad, just the fact. And often I'd read books and fantasize about, oh, why can't I have this life? And I feel like, well, my life, I compare it to a lot of things that were different than mine, but that made it for me seem better than it actually was. And so I think that it was just really, really hard. And thank goodness my parents recognized it. Otherwise, I wouldn't be here in front of cameras.
Yeah, I mean, we were actually worried about, things like suicide in kindergarten and first grade. It was terrifying. Yeah, and we're not really worried about it. I should say I don't want to exist. I wish I didn't exist. And we have a lot of kids, so we're not really alarmist. Yeah, a bomb has to go off to really phase us. It was terrifying. Yeah.
I wouldn't want to hurt myself or die because both of those things scared me. But I did want to just not exist, like poof into magical fairy dust air and never have to face anything again and be happy.
Ned, how old is she?
I think she was about 9 or 10 in this. I forget.
Well, she is really articulate. Are all the kids in Madison that educated and articulate?
No, not all of them are. She's very, very talkative as well. Can you hear me okay?
Yes.
Okay. Well, I'm glad we could get that to work. Thank you, Simon and Jeff, for that. So that gives you a feel for what this is like at a young age, all the comorbidities and also the suffering. One of the landmark studies that was done and published in the New England Journal a number of years ago was a study that examined the treatment of anxiety disorders in children using a comparator between CBT, sertraline, and the combination. And it was a sort of a landmark study in a relatively large group of kids demonstrating that the combination, maybe not surprisingly, of medication and psychotherapy was effective. About an 80% were improved on the CGI scale, as compared to 50% or so with the individual treatments, as compared to a relatively low placebo response at around 20%. So this is the good news. The good news is that there are treatments that work for kids, and certainly treatments are evolving. The not so good news is the follow-up of these children, and this is a complicated paper, but the naturalistic follow-up of these kids 4 to 12 years later, basically following them for 4 years, suggested that only about 20% of the kids stayed stably remitted, 30% remained chronically ill, and 48% relapsed. And so while we have good treatments in the short term, the challenge, of course, for this illness and other illnesses that we treat is to keep people better and to actually do something that will have more of a permanent change from the standpoint of their resilience. And that sort of leads to this whole idea of neuroplasticity and changing neuroplasticity, which I'll come back to in some of our non-human primate studies. So I've already made the point that early life risk is important. I don't have to convince any of you about that, I don't think. But this is just to show you sort of the relative age of onset of many of the disorders that we work with, illustrating the fact that anxiety disorders come on early. Depression usually comes on somewhat later, and many people think that early anxiety is frequently a forerunner of adolescent depression. And one of the studies that we're involved with is following young girls, as I mentioned, who have high levels of trait anxiety, who do not meet criteria, and actually trying to understand what their longitudinal course looks like in relation to their brain development from the standpoint of the circuits that we're most interested in. Now, one of the phenotypes that has been well characterized to be a risk factor for early life anxiety disorders and later depression and even comorbid substance abuse is something called behavioral inhibition. This was a term that Jerry Kagan, a famous psychologist at Harvard many years ago, basically explored, demonstrating that young kids could be classified as being extremely inhibited in novel or unfamiliar situations or when approached by strangers, sort of like extreme, and I mean extreme shyness, and that kids that had this predisposition tended to stay stable with this predisposition as they developed. And other researchers, including colleagues at Harvard and MGH, later elaborated that these children were more likely to develop anxiety problems and other stress-related problems. In a meta-analysis by Jenny Blackford at Nashville, her work has demonstrated basically collectively that there's roughly a 3 to 4-fold risk over the general population, which already is, the incidence is fairly high, of developing social anxiety disorder and other anxiety disorders. And importantly, we've been able to develop a model of this temperamental risk in non-human primates as a way to begin again to study human risk that we think is a very, very good predictor of later life psychopathology. This is just showing the meta-analysis from this study. So what I'm going to do now is I'm going to play you another video, this video is from another study that we've been engaged in over the years called the Wisconsin Family Study, where over 500 families have been followed with their children prior to birth in moms, and then all the way up now through late mid-20s. And this is a video clip where you're going to see roughly a 9-year-old child, one that is tested in his home, asked to give a little speech and is being monitored physiologically from the standpoint of heart rate and autonomic reactivity as well as cortisol. And then he is selected for being on the uninhibited spectrum of this behavioral inhibition. There's a range, obviously, of how inhibited people are temperamentally. He's selected to be on the extreme uninhibited side. And then I'll show you his counterpart who's been selected to be on the inhibited side, just to show you what this temperament looks like at this particular age. And let me make sure that I have this, get this set up right here. So let's try this again. So we're going to go to panel speakers. Okay, make sure the volume is up. All right. Okay, so let me know if you can hear this, Simon.
No.
Can't hear it? No?
There we go.
Straight. You'll have 2 minutes to give your speech. When I tell you to start, I want you to tell us about.
Can you guys hear that?
Yeah, yes, it's okay now.
Okay.
Day yesterday. You can stop when this timer runs out. Tell us about your day yesterday.
We're not hearing very much now.
Okay. So let's, you know what? Let's just take a, let me,
Tell us about your.
so can you hear me?
Yes.
Okay, just take a look at the second kid here. You can see he has no eye contact and he's not moving at all. Can you guys see that?
Right,
And,
right.
you know, basically he's the inhibited kid. You saw the other kid that was wiggling around and smiling. You couldn't hear him. He was talking. This one isn't talking. If we were to measure physiology, this child would probably show high levels of autonomic activity. If we were to look into his brain, we'd probably see activity in regions that are related to the neural circuitry that has to do with fear and anxiety, including the amygdala. So I'll move on then. The idea here is that basically that people more or less come into the world with different ranges of temperamental anxiety, ranging all the way from low to extremely high. And that if you're in the high range, but subclinical overdevelopment, the 3 trajectories that are possible are that you'll, with stress or some other reasons, to start developing a disorder. You may stay stably high, but not meet criteria for a disorder. And in some kids, interestingly enough, roughly 25% seem to get spontaneously better. And so what we're doing is we're really interested in trying to understand what the sort of neurodevelopment is that underlie these 3 trajectories. with the hope that by understanding these different outcomes over time, we'll gain insights into what we might think about doing earlier in life. Now, I mentioned that I'm very involved with working with non-human primates, and the reason that we work with rhesus monkeys is because they're old world monkeys, and they share many features with humans from the standpoint of their social behavior, from the standpoint of their early nurturance of their young, from the standpoint of the stressors they face, which are primarily social, and also interestingly from the standpoint of that they read and respond to visual cues related to facial expressions and to threat, which I'll show you in a minute. If any of you have visited Lake Mendota, or visited Wisconsin, we have a bunch of lakes, including Lake Mendota. This is not the monkeys from our primate lab posing in front of our lake. This is in Puerto Rico, off of Puerto Rico, a small island for many years, where we had a research project that I had the great fortune of being involved with, especially in the winter months, of getting a chance to go down there and to work with monkeys in a more naturalistic and wild situation, if you will. What we've done over the years is developed a model that we believe captures what you just saw basically in those two children, and what I'll show you a little video of a young toddler in a moment that has behavioral inhibition that is extreme. And basically what we're measuring defensive responses or fear-related responses that are adaptive from the standpoint of survival. And in this case, we're very interested in freezing behavior, which is an adaptive response, which I'll tell you a little bit more about in a moment. And we're also interested in the reduction in verbalizations, because inhibited children and people basically are fearful of situations, they're avoidant, they tend to be worried about performance, and they tend to have a longer latency to speak and speak less. And so we have a combination of increase in defensive behaviors, freezing, a decrease in vocalizations, and we also have thrown into this physiological reactivity, which is the threat-related increase in cortisol. So we've expanded the human definition of behavioral inhibition to include a physiological measure, and we actually believe that the inclusion of this physiological measure, cortisol, actually may enhance the predictive value of this type of phenotype and construct. And so we've termed this anxious temperament. It's a combination of these three indices, and we've shown in the wild on this island of Cayo Santiago that in the laboratory, if we assess an individual's level of anxious temperament, it's predictive of how much social interaction that individual has in the wild, and in young monkeys, how far they venture away from their mothers. Now, I'm not going to go into how we developed all this, but suffice it to say that many years ago, through my interest in relation to stress and stress reactivity, I was able to come up with some ideas about how you can elicit these types of responses simply by changing whether or not an individual has eye contact with a monkey. And so there are three conditions that elicit different responses, all of which are stressful and all of which call for different defensive adaptive responses. If a young monkey, as Harry Harlow described many, many years ago, is taken from its mother for a brief period of time, or a young human, what they do is they engage in calling for help behaviors or crying behaviors. That's a signal to come get me, come retrieve me. I'm scared, I need support. However, if there's a threat in the environment, it's not adaptive to be making lots of noise and drawing attention to yourself. If there's a threat in the environment and you believe that the threat has not yet found you, then your response is to stay quiet and to freeze in one position and to stay very still, to try to remain undetected, and while you're trying to figure out what to do, to problem solve, to escape. And this is elicited in our paradigm by a person coming into the room and only presenting his or her profile and not having eye contact with the monkey. And this is very critical. This is the condition that we use to phenotype the monkeys, and we think it's very similar to what is done in humans with what we call the stranger situation. And then finally, if you stare directly at the monkey, you get yet a very different response, where the monkey quickly shows you its weapons, these big canine teeth, and gives you a sense that if you mess with me, I'm going to either fight back or in some cases run away. And so simply by modulating eye contact, we elicit these very different responses that are adaptive. And this is one of the reasons the primate model is so valuable, because we know that eye contact is so critical in humans. Now, all of you that have children or had children probably recognize or remember this wonderful book by Maurice Sendak, Where the Wild Things Are. And the reason I'm showing you this is because it really nicely illustrates the point that I'm making about the importance of eye contact in social communication, and also in eliciting fear-related responses and neural activity-related fear-related responses. Also, this dawned on me when I was reading this book for about the millionth time to my oldest son when he was a young boy, and I was doing this research. I was very enamored with the idea that I had come up with, this idea about eye contact and how important it is, and so on and so forth. And then after reading this book, it dawned on me, and I realized that everybody sort of knows all this stuff, and we know this intuitively and sort of unconsciously. But if you remember the story, Max is a little boy. For whatever reasons, he was misbehaving. He got sent up to his room, and of course, in the psychoanalytic tradition, he had a dream. The dream was that he got on he basically got on a ship, and he sailed across the ocean to an island. And when he arrived on this island, this is what Maurice Sendak says, and when he came to the place where the wild things are, they roared their terrible roars and gnashed their terrible teeth and rolled their terrible eyes and showed their terrible claws. Till Max said, "Be still," and tamed them with the magic trick of staring into their yellow eyes without blinking once, and they were frightened and called him the most wild thing of all. So we all know that eye contact is critical, but for anxiety patients, it's even more critical sometimes from the standpoint of avoiding eye contact. And also, it is a cue and a signal that actually elicits amygdala activation and activation of other subcortical structures that are related to fear and anxiety. So what we've tried to do is to, again, as I've said, is to translate back and forth across species. In humans, one of the things that we do is we have a stranger come into the room, and we look and measure the physiology, and we measure the behavior of the young child and can phenotype the child along this continuum of behavioral inhibition. In the monkeys, we do a similar thing where we have a person come into the room, stand and present his or her profile. We measure freezing behavior, reductions in vocalizations, and in cortisol. And what I'm going to do now is show you one more last video clip. I won't play the volume, but I just want you to look at this. On the left is a monkey that's been selected for being very extreme in its anxiety and its inhibitory response. And on the right, as you look at it, will be a young child who's with his mother, who's being tested in one of our studies for temperament. And he responds in a dramatic way as well from the standpoint of his inhibition. And both are selected based on their extremeness in this dimension. So you won't be able to hear it. Suffice it to say that the mother and child are talking. So if you watch this, you can see the monkey's walking around and the child is playing with the familiar graduate student. The mom is sitting in the corner here. If you could hear it, you'd hear a lot of talking. You see the little child is smiling. Monkey is making some calls. This is a pre-adolescent monkey as well, a younger monkey. And in a moment, you'll see what happens when the intruders come into the room. So there's a knock on the door, there's sort of a queue, and things are still fine. Now look what happens to the monkey over here when the intruder comes in and presents his profile. And over here, you can see that there is a reduction in movement. This monkey is freezing in one position. And as you watch this young child, the child gets stiller and stiller and quieter, stops talking to mom, stops playing. Many children will run back to mom. Many children will start to cry or say something. And bold children will actually engage the stranger in that regard. And the same thing with monkeys. We see this very wide range of behaviors. These have both been picked for being extreme. If we were to measure cortisol or brain activity, we would see definite changes in relation to this. And I won't belabor this, but there's a time lapse here that goes on, and this child is still sitting here, not moving, not having eye contact for about 9 or 10 minutes. And the same with this monkey. When the intruders leave, everything goes back to normal.
Ned, when you say these were extreme cases, these were extreme examples of intruder anxiety or reactivity?
Yes. These are selected for being extreme from the standpoint of their inhibitory responses, their anxiety responses. And that's the phenotype early in life at when it's most extreme. But the point that I want to make is that's the extreme example. There's a broad range of reactivity from the standpoint of temperament. And what we've been doing is trying to understand the individual differences and how individual differences relate to brain. So you all know about the amygdala. I won't belabor that either, but work by Joe Ledoux and many others in rodent species has allowed us to understand the subcortical machinery that mediates our fear and anxiety responses as well as other motivational responses. The amygdala is not just dedicated to fear, it does lots of things. But what's really interesting about this response is that it can be so rapid that we can actually bypass sometimes the conscious recollection or the conscious acknowledgement of what's triggering some of our responses. So the point to make is that the amygdala is critical. It's in the medial temporal lobe. The amygdala is a complicated structure. It's not one thing. I'll get into that a little bit later. But most importantly about this is in primates, the well-developed prefrontal cortex that you see here in a rhesus monkey that is analogous to a well-developed prefrontal cortex in humans is bidirectionally connected to the amygdala from the standpoint of the ability of these two regions to talk to each other. Most of the connectivity, as you can see, is around sort of the pregenual and orbital frontal cortical regions, posterior orbital frontal cortical regions, as well as some of the cingulate regions. And as the point that I made is that these are bidirectional connections. This is probably how we impact our stress reactivity and anxiety levels through prefrontal interventions like perhaps CBT or psychotherapy or TMS. And it's also how we can get ourselves very worked up from the standpoint of our worries with activity in the subcortical regions communicating with our prefrontal regions. But most importantly, and the point that I want to underscore is that the primate model is particularly well suited for this because of the well-developed prefrontal cortex as compared to rodent species. And I'm just going to review for you now, because of time, pretty quickly, some of the studies that we've done across children, pre-adolescent kids, and pre-adolescent monkeys that demonstrate that not only is the phenotype similar at a behavioral level, but also when we begin to look at brain function, we see similarities as well, which really validates the model we use and allows us to gain some insights into human anxiety that we otherwise probably wouldn't have. So as I mentioned, the amygdala is a complex structure. There are nuclei in the basal ventral regions. In the dorsal regions is a structure called the central nucleus of the amygdala. The information from the cortex and from other subcortical structures and from motor and visual cortex and prefrontal cortex basically enters the amygdala where it's processed and learning can take place in these basal regions, and the outflow to autonomic regions, to motor structures, to behavior, to our adaptive responses flows out of these more dorsal regions, the central nucleus, and also another part of what's called the extended amygdala, the bed nucleus or the stria terminalis. In a study that we did a number of years ago with Lisa Williams in our group, we were able to show that when we looked at children who had anxiety disorders, these are pre-adolescent kids, and we put them in the scanner and did a paradigm where we could vary the level of whether or not individuals were going to see a negative picture or a negative face. There were three conditions: a specific cue that's a red X that said, okay, this is going to be followed by a negative face all the time, a blue N that said, okay, this is going to be followed by a neutral face all the time, and a question mark where it could be followed 50-50 by either one of these. What we found was that the question mark situation, the uncertain situation, was a situation that revealed differences between anxiety disorder kids and controls, with more reactivity in the amygdala region and also across sort of the brain in many regions, including the anterior insular region. So this is just a demonstration that in the scanner we can see changes between kids that have anxiety and those that don't. And interestingly enough, and not surprisingly perhaps, uncertainty is a set of conditions that particularly activates this region. Now, in an analogous fashion, but a little bit different, we've studied a large cohort of monkeys, almost up to 600 monkeys, from which we know their genealogy and their background and their family histories. And we actually did PET scanning, which is we're using FDG glucose, where we can look at brain metabolism, which is a bit of a different measure than looking at the bulge signal with fMRI. But in these close to 600 animals, what we've done basically have been to phenotype them, and at the same time, when they're being phenotyped, to administer this radio tracer that acts as a glucose analog so that we can quantitate brain metabolism at the very same time that we're quantitating individual differences in behavioral response or in anxious temperament. And so then what we can do in our analysis is we ask the question, how do individual differences in anxious temperament all across the range from low levels to high levels and middle levels in between relate to individual differences in brain metabolism? And what you're seeing here in red are the areas that are highly significantly predictive of individual differences in the phenotype. What's really interesting is that in the middle here, you see that the amygdala is highly predictive, especially the central nucleus region, this dorsal region. But of course, it's not just the amygdala, it's across the neural axis in the brain. Up here, these are posterior orbital frontal regions, some of the regions that are connected to the amygdala. Up in this region is the bed nucleus stria terminalis, the anterior hippocampus, and interestingly enough, the strongest signal was seen in the brainstem in the periaqueductal gray region, which is a region that has to do with freezing behavior from studies in rodents, and many studies in humans have also documented that this region is very much involved with pain processing. I mentioned already that we have the pedigree. This is not the wiring diagram of the New York subway system, but basically if you look at the colored circles, these are all the individuals that we studied with imaging, but all these connections are the generations that we know how they're connected. This cohort is particularly well constructed to do genetic heritability studies because there are many, many half-sibs in this cohort. This allowed us to estimate the heritability of the temperament, anxious temperament, to be around 30 to 40%, which is entirely consistent with what we see in human anxiety disorders. But more importantly, and I think more in a novel way, we're able to also ask questions about how heritable is brain function in the regions that we're interested in. And so this cohort allowed us to basically look at degrees of heritability. In yellow, what you see outlined here are the regions that are predictive of individual differences in anxiety or anxious temperament. But in green are the levels of heritability that you can see on this scale over here. You can see that virtually every region that we identified as being predictive of anxious temperament is also significantly heritable, although the heritability levels range from very, very low up to about 50%. But heritability per se is not the most interesting question. The more interesting question is, what is the co-heritability of brain function in relation to the phenotype, the behavioral phenotype or anxious temperament? And basically what that question is asking is, which of these regions have the exact same pattern of heritability as does the phenotype if you follow it through the family tree? It's entirely possible for these regions to be heritable, but not heritable in the same way that, for example, anxious temperament is heritable. And so by doing an analysis that looks at genetic correlations, we can ask the question, what are the brain regions that are co-heritable with anxious temperament? And when you do this, what you see is really only 3 regions that are significantly heritable in the exact same pattern that anxious temperament is: the posterior orbital frontal regions, this area called the bed nucleus stria terminalis. Interestingly enough, the amygdala is not co-heritable, suggesting that their environmental influences may be more prominent here. And again, the periaqueductal gray region. This also suggests that the same genes that are involved with the behavioral phenotype may be involved with the expression, a regulation of these brain regions in relation to threat responsivity. Now, we've also done fMRI in the monkeys using resting state or so-called resting state, have been able to demonstrate changes in connectivity, functional connectivity, resting connectivity in the children that we studied between the amygdala, the dorsal amygdala region, the central nucleus, and the dorsolateral prefrontal cortex, with less connectivity being associated with greater levels of anxiety, and we're actually able to reproduce the same exact finding, or more or less the same finding, in our cohort of rhesus monkeys. So in addition to these regions being metabolically different, we can begin to see data that supports alterations in the prefrontal amygdala or prefrontal sort of a subcortical regulation of these structures in relation to anxiety, with the idea being that this mismatch between prefrontal input and cortical input and output and amygdala regulation may be a mismatch in relation to what's going on in stress-related psychopathology, especially anxiety disorders. We can also use DTI, as you know, to get a proxy of the integrity of white matter tracts, including perhaps myelin. The unsina fasciculus is a region of particular interest to us because it is a pathway that connects the medial temporal lobe, including the amygdala, to these regions of the prefrontal cortex, especially the orbital frontal regions. And in a number of studies led by one of our graduate students, Do Trump, we were able to show that the integrity of this pathway seems to be decreased if you use measures of fractional anisotropy in relation to adults that have anxiety disorders, children that have anxiety disorders, and now we've been able to show this more recently in non-human primates. And interestingly enough, this appears to be a sexually dimorphic finding, at least in earlier ages. We found this to be the case mostly in boys, and to be less the case in girls, both in our monkey population and in our children, in our young girls and boys. So how do we move beyond this? So again, to remind you, the way we're thinking about this is that there's a whole series of structures that are involved in regions in modulating anxiety. We have conceptualized this as has the field, that the sort of the extended amygdala region is the core of this circuit, perhaps, with outputs to the brainstem and inputs from orbital frontal cortex, modulating the amygdala function. And the value of the non-human primate model, of course, is that you can, or any animal model, is that you can do mechanistic studies. Many years ago, what we did is we went in and inactivated this region of the amygdala that is important. We believed it was important, that had been done in rodent studies. And basically, by using a neurotoxin to kill the neurons in this region, we're able to show reductions in freezing behavior and changes in cooing and ACT and cortisol activity, all of which are consistent with this region being involved in anxious temperament, such that when you reduce the function of this region with a lesion, you basically transform monkeys from being more anxious to being less anxious. Note that the reduction in freezing is significant, but not dramatic. And I think that this really relates to the idea that we're not just talking about one structure being involved in these complex behaviors and phenotypes and syndromes, but rather the whole brain with a lot of redundancy in structure and function. Now, from the rodent studies has emerged really elegant strategies to manipulate specific molecules and circuits. Chemogenetic strategies and optogenetic strategies, many of you have heard about, are ways to have greater precision in asking mechanistic or hypothesis-driven questions about causality. We elected to use a model or a system called DREADS, which is actually a very appropriate term, I guess, for psychiatry. But basically what this does is it employs a mutated cholinergic or muscarinic receptor that will only bind to a molecule that is relatively inert. In this case, clozapine oxide. This is a whole other story. I won't get into the details of this about the ligand that's used. But the way that this works basically is that you can genetically manipulate an animal by using a viral vector to have cells express these mutated receptors. They will only recognize a specific molecule that you give as a drug. And by so doing, you can either activate or inactivate the receptor and modulate the firing of that particular neuron or cell. So we've spent a lot of time developing methods in monkeys where we can use viral vectors to actually over or underexpress specific genes or molecules. We were fortunate enough to be able to work with a group in medical physics to develop an intraoperative MRI strategy where we can sort of more or less in real time visualize our target, in this case, the dorsal amygdala, can in real time infuse through a small cannula the viral vector that contains whatever gene it is we're interested in, and at the same time use some gadolinium as a imaging agent so that we can actually see where the infusion is. So with this kind of technology, we've been able to use what I already mentioned was this DREADS strategy as a chemogenetic strategy. Here's an example of an infusion into one side of the brain in the amygdala in part, and then later, 3 or 4 weeks later, after the gene has been expressed, we can actually use a ligand that will bind to this receptor and show that the receptor is being overexpressed in this particular part of the brain.
That's muscarinic receptors?
That's a muscarinic receptor, yeah. So the idea here is that in addition to using the non-human primate as a mechanism, a way to understand mechanism, we also believe that this is a really important platform to think about proof of principle and concept studies for humans, where we think many of these strategies actually may be applied in neuropsychiatric problems, neurodegenerative problems, and even in, I would say, severe patients with psychiatric disorders are not out of the question as we think about the future of some of these technologies. This is just to show that this molecule is overexpressed in the amygdala, and with electron microscopy, we were able to show with a collaborator that it's expressed on the cell surface or the synaptic surface, so that this is something that can be reached by molecules that are administered. And to make a long story short, in an initial study that's now being submitted to a journal, we're able to show with this technology that we can reversibly basically make animals less anxious by inhibiting the neurons with this technology in the amygdala. When you give the external agent, in this case, it was very low doses of clozapine, but you can give other things. And when you turn these receptors on, they inhibit the function of the region we're interested in, and it resulted in a reduction in anxiety. When you don't administer that drug, the animal goes back to its regular state of anxiety. Further proof, basically, that this is a system that can be used, as well as a circuit that may be an important target circuit. We're doing studies in the orbital frontal cortex as well, and also trying to now look at very specific pathways with other technologies. Now, the other advantage, of course, is that you can do studies that look into brain mechanisms per se. And we've been able to develop actually a brain bank over the years of animals that have been phenotyped for anxious temperament. And based on that, have been able to look at very specific brain regions or even neurons now that we're interested in to try to understand what the gene regulation looks like in those particular areas. And so what we've been most interested in studying is RNA. And the reason for that is because RNA, as you know, is the intermediary between our structural genetic variation and the proteins that are so critical from the standpoint of function. And RNA is also really important because it not only reflects structure of genes, it also reflects epigenetic factors that may be related to non-genetic environmental types of effects. So in an earlier study, what we did is we actually punched out the region of the brain that we know is so critical from the standpoint of its metabolic function in relation to anxiety, and then characterized individual differences in gene expression in relation to individual differences in temperament. More recently, we've used technologies where we can actually go in and with a laser capture individual neurons. And so this now allows us to not be looking at other things besides the neurons that we're interested in. And as some of you may know, if you're involved with the science side of all this, there are now technologies that we're using where we can actually look at individual neurons one at a time, in a sense, from the standpoint of their gene expression. Our collaborators, Jim Knowles and Tade Sawawe at the time, were instrumental in this. And to make a long story short, what we did is we searched for RNAs or gene products that were predictive of individual differences in anxious temperament. So we're looking for, can we find genes or gene expression that is lining up with high, middle, and low levels of anxious temperament? And one of the things that we found was that neuroplasticity genes seem to be a particular class of genes that were related to anxious temperament, which is interesting because of all the work that's been done in the hippocampal region in relation to neuroplasticity. Less work has been done in the central nucleus of the amygdala in relation to neuroplasticity. And interestingly enough, what we found is that individuals that were higher in anxious temperament actually had lower levels of gene expression for a variety of neuroplasticity genes. The converse was that animals that were much less anxious had higher levels of the activity or regulation of some of these genes. One of the genes that we became very interested in is a gene called NTRK3. And this is a gene that is analogous in a sense to other neurotrophic receptors. So the receptor for BDNF, for example, is in this family. And the ligand for NTRK3 is a molecule that is sort of similar to BDNF. It's called neurotrophic factor 3. Anyway, we found that individual differences in this gene expression were related to anxious temperament. And as you can see here on the y-axis over here is individual levels of anxious temperament, and on the x-axis are individual levels of gene expression. And you can see this inverse relation such that higher levels of anxious temperament are associated with lower levels of this particular gene and the activity of this gene. This is the pathway. This particular growth factor turns on one of these tyrosine receptor kinase receptors. In this case, it's NTRK3. There's a whole cascade of intercellular changes, including other members that we found of this cascade that were also altered in relation to anxious temperament. So, to test this hypothesis, and again, this is an association. The question is whether or not we can really show whether this particular pathway is involved. To test this hypothesis, what we decided to do was to overexpress the ligand that interacts with this receptor. If the receptor is down regulated, our hope is that we could actually increase function of this pathway by increasing the ligand, the endogenous ligand that activates the pathway. So this is a way now of combining sort of these manipulation studies that I told you about in relation to a circuit with information that we understand about, that we think is related to the molecular underpinnings of the function of the circuit, and then actually to manipulate this with the idea that this could bring us some thoughts about new drug ideas. So again, the idea here was that this particular receptor is downregulated in individuals with higher levels of anxiety, at least its gene expression is, and our idea was to go ahead and put a viral vector in this region that would make more of the ligand that turns this system on, with the idea that this should be a way of reducing anxiety with a molecular manipulation of cells or neurons in a brain region that we've already characterized to be important in mediating anxiety, or a circuit that we've determined to be important. This is just an example again of putting this into this region. Over here you can see these are the 5 animals in which there was where we infused it, and this is the overlap of the infusion if you look at this with gadolinium. Make a long story short, again, we were able to show that we could get a significant reduction in anxiety simply by increasing the gene expression in this very specific region of this endogenous molecule that interacts with this receptor. So this is proof of concept then that we can manipulate specific genes. And again, the value of this is we could also image the brain in these animals, and we're able to show that where we manipulated the gene expression, we also saw some significant changes in metabolism when these animals were in a threatening related position. So to sort of wrap things up, what I've tried to do is to sort of give you a broad overview of how we can go from understanding the importance of human symptoms and syndromes to thinking about development and ideas about early detection, early intervention, especially in this case in relation to anxiety, how we can think about a phenotype that may be a risk phenotype in humans, really reliably model that in non-human primates, capitalizing on the value of the non-human primate model from the standpoint of its evolutionary relatedness to humans, and then further dive deeper to understand circuits and molecules in ways that hopefully will shed light onto new ideas about how we think about human illness and also hopefully human intervention. This is the large team that I'm extremely grateful to who have been involved with this research over the years. It really has truly been a very, very much a team effort, and a pleasure for me to be able to work with all these folks and to collaborate. This is a springtime day in Madison with my lab. So thank you for your attention, and I'd be happy to entertain any questions that you have. Thank you.
Terrific. Thank you so much, Dr. Kalin.
Sure.
That was a terrific talk. I was going to start, I guess, with a comment and then a question. So the comment is just both the breadth and the depth of your work is really remarkable, going from human studies of temperament all the way to viral vectors in animal models. So really elegant work, and thank you so much for presenting it.
Thank you.
I guess the first question that I had is, as a child psychiatrist, I really appreciate your focus on development, and I wanted to go back to the camel study that you mentioned, the follow-up on the study from the New England Journal by John Walkup, essentially showing that in spite of an initial quite a good response to treatment, over the long term, these kids did not do very well. And those findings are actually quite similar to what the MTA study showed, the multimodal treatment of ADHD, where initially the kids responded quite well to treatment. Over the long term, they really suffered. And it's quite disheartening for a child psychiatrist, where we believe that if we identify these kids and we intervene early with effective treatment, we can change the long-term course of illness. And so I guess my question for you is, what are we getting wrong, and how should we think about the long-term trajectories of these illnesses?
That your question is the holy grail, basically, of what we're after. And my own sense is that that we have to understand, sort of as I've been talking, the early circuitry and how that circuitry is modulated by both environment and genes, and then really have interventions that are impacting the circuitry well before this sort of more chronic pattern of behavior or emotion dysregulation is instantiated and maintained. And that's why I think that's why we selected the neuroplasticity sort of side of things, with the idea being that this might be an opportunity to think about how to modulate and affect a change that would have sustaining effects, or would change a trajectory developmentally. And it's also, I think why we've taken the strategy to follow children longitudinally and to try to understand different trajectories and what's going on neurodevelopmentally in relation to that, with the hope that kids that have better outcomes, that we could learn something from them. It's really interesting to me that 25% of kids that look like they're as affected as other kids seem to get better over time naturalistically. And what is that? And I also don't want to limit my point to a biological or to simply a sort of a within brain point. Early environment, family interactions, parenting, nurturance, all that obviously is critical as well. And how those things impact some of these target circuits, I think is going to be really important to understand. But one of the things, another thing that I've gotten very interested in, and actually I'm writing a grant on now, or trying to, is the role of sleep in all of this, especially during development. And for sure, anxiety disorders are associated with a lot of sleep disturbance, and children that have anxiety disorders have a lot of trouble falling asleep and staying asleep, and pre-sleep arousal. One of the things that came out of that camel study, I think, and other studies, is that kids can get better from the standpoint of anxiety, but frequently the sleep disorder is maintained for the sleep disturbance. So it's just another example of, how broad some of these phenotypes might be and how different physiological components may be impacting things.
Dr. Lieberman, do you want to follow up with a question?
Yeah, but I see that Jeff Miller submitted a question on the chat function. So he compliments you on your talk and says, given the pharmacological interventions in humans generally hit everywhere in the brain where the target is expressed, do you have thoughts about how to translate the viral vector studies from non-human primates into humans, and how to get both anatomic and chemical specificity?
Yeah, so I have a lot of thoughts about that. And I do believe that we do we will have the technologies, I think, to both have circuit-based and cell-based specificity in humans if we want to pursue that. And I say if we want to pursue that, obviously, because of the invasive nature of thinking about gene therapy in a sense, or the manipulation of cells in this very specific way. However, given everything that we've already are doing, including deep brain stimulation, lesioning in refractory patients, and so on, and also neurology is moving forward with trying to use these strategies in some severe cases of neurodegeneration or epilepsy. I think that this is a viable pathway, we believe that you can really get down at the cellular level. One of the problems previously has been making these viral vector, making the constructs that go into the viral vectors and the viral vectors themselves having enough selectivity to express some subpopulations of neurons that are specified. And the rodent studies have moved, with optogenetics, are clearly have moved to that point and beyond. But the chemogenetic studies, like the ones I'm talking about, have been more difficult. But we've been working with collaborators now that have very selective promoter regions or enhancer regions that are linked to the constructs of interest that allow, for example, very specific expression in GABAergic neurons, or even more specifically in subtypes of GABAergic neurons. So the technologies are there. I think the question is how and when do we begin to think about translating this to humans? But one of my goals with the non-human primate work is to really develop some of the potential methods and understand the pitfalls that may be there when we do actually think about translating this to humans.
Jonathan, if I might follow up with a question of my own. Actually, it's two related questions. First, in the context of the COVID pandemic, we've seen a lot of individuals have exacerbations of their mood anxiety symptoms, and also even to the point of elaborating kind of phobic delusions of contamination and so forth. So, the question is would you hypothesize, or would you conclude that individuals with the kind of temperaments that you've been characterizing developmentally are those that may be vulnerable to react so extremely? And then related, how do you integrate your model developmentally of temperament and sort of circuit-based reactivity into what we call borderline personality disorder, or the induction of PTSD on environmental traumas?
Okay, so good question. So COVID, I think the immediate response to your question, Jeff, is yes, you would think that these individuals would be more vulnerable and more reactive. And they may be, but some of the things that I've been hearing anecdotally, as well as through my role in the journal with some things that have been submitted, have been the surprise that some people with anxiety, maybe not a surprise, feel a lot better because if they were already obsessive, they know that they were sort of doing the right thing, or maybe they were right if they had fears of contamination. If they were socially phobic, there's less of an imperative and almost permission to not go out of the house. And so in some sort of paradoxical way, there may be some symptom reduction in individuals that have pathologies that sort of fit the context of the changes that we're making. By the same token, all of us know that people who tend to be less inhibited and a little bit more extroverted may be feeling more distress in relation to not being able to get on an airplane. As a personal example, I'll reveal a fantasy that I had the other day, which struck me as being at first rather odd, which was that I was fantasizing about being in the Delta Sky Lounge, which I used to hate because I was always associated with waiting for flights. And now I was thinking, gee, wouldn't it be nice sitting in there with nothing to do? But anyway, so I think it's it's this interaction between temperament and personality and context that sort of shapes the symptoms that that one is having, or how important one is valuing those symptoms. So I'm sure many people are more reactive during this time, but it may not be the suspects that we initially think about. That doesn't mean that they're not suffering. It doesn't mean that they still aren't, they still don't have their problems. It just means that the ego dystonic aspects of their symptoms may not be as apparent to them. The second question was about sort of PTSD and borderlines, and how do I think about the developmental sort of ideas in relation to that. And again, I mean, for me, it's not that different, I guess. I think that, we tend to think both of the examples that you used are very traumatic related examples, whether it's early life trauma related to borderline development and, mixed up messages with parenting that are severe, or later trauma related to PTSD. Again, I think there's a vulnerable substrate, and I actually believe that the anxious temperament that we're talking about is more or less there at birth. We've done studies now in newborn monkeys showing that we can measure amygdala metabolism under somewhat stressful conditions in a 2-week-old monkey, and that's predictive of amygdala function at about a year of age, which in a monkey is probably like a 4 or 5-year-old child. And so I think that, these are very modifiable circuits by environment, but they're the sort of genetic, biological early predisposition is there. And then on top of that, overlaying the experiences of life and environment, and when those are extreme and when they come together with the right disposition, I think you see a borderline person, versus somebody else.
Great. So we don't have too much more time. I just wanted to make one comment and then add another question. The comment was, I guess I would be remiss in not mentioning this, that the fact that something is present from birth may suggest that it has a genetic underpinning, but certainly doesn't lead to that conclusion. That certainly there are pretty facts that are important.
Absolutely. Thank you for that clarification.
The question, which I think you may have touched on a bit already, comes from Kevin Bath, and it is for the NT3 TRK-C studies, does the age at which the AAV NT3 is delivered, does that matter?
That's a great question, and I wish I could answer it. Because these studies are so complex and take so much time and are so costly, we've had to limit the number of subjects that we use, and can't ask those questions yet. But it's a great idea for a grant, so thank you. I would think that the earlier that we could make these manipulations, theoretically, the more of an impact they might have over time. So it would be very interesting to do this in much younger animals. The animals that we've been focusing on have been pre-adolescents, mostly to try to move back and forth between the children that we're working with. But going earlier would be really interesting.
Just to follow up on so you describe a lot of different options or possible strategies for treatment development, and I suppose you could include TMS, targeted TMS with those. But at the same time, clinically, for years, the Procrustean modalities, pharmacologically speaking, have been benzodiazepine targeted treatments or serotonin reuptake inhibitor treatments. And yet, you were using clozapine as a muscarinic agonist, and you've identified various molecular targets that could be chemogenetically probed. I mean, how is this going to? How do you see this getting translated into treatment development?
Well, again, I mean, I wish I could, have clear pathways. But so one idea is that, let's say that we identify and we're confident that the findings that we have are going to be useful. this would suggest that the central nucleus region of the amygdala, this dorsal amygdala region, is a key site. It would also suggest that increasing neuroplasticity in that region could be a key modulator of anxiety responses. And so then the question becomes, well, how can you get to that site and increase neuroplasticity early enough in life, as was already pointed out, that would make a difference with a trajectory of a development. And here, I think simple things like, exercise, for example. We know exercise increases neuroplasticity. We know it increases BDNF. I don't think anybody knows what it does to this system per se. It's a very related system. I'd be surprised if it was different. And I don't know if anybody knows what it does in the region of the brain that we're interested in. Is there a critical period in a child's life where if somehow we stimulated, the plasticity of this system, that it would make a difference? Are there ways that we're sort of already have in our hands that are sort of common sense ways that if we did it at the right time and with the right dose and the right person, that might make a difference? It seems simple, but those are the kinds of other ideas that I've been thinking about. And again, it's not, I think the other point here is it's not just one brain region. And I think that, while this talk is very focused on a region and sort of very reductionistic in a way, another takeaway point that I really want to emphasize is that from our monkey studies and our human studies and from the literature, anxiety is associated with brain-wide alterations in the function of the regulation of anxiety. And this is all the way from prefrontal, dorsolateral regions to orbital frontal regions and ventromedial regions to, anterior hippocampus, extended amygdala, some medial dorsal thalamus, periaqueductal gray, other brainstem regions involved with catecholamine modulation, and so on. So while we've poo-pooed our sort of treatments that hit everything, part of the reason that they may be effective from a neuromodulatory standpoint is because they do hit everything or hit this broad range of things. And so it'll be really interesting to see if you have a very targeted treatment, if we can get there, is that really going to be fundamentally effective in a way that's different than some of these broader treatments? And by the same token, you can think of, we haven't investigated this NTRK3 pathway in other brain regions yet. So is this something that's specific to the central nucleus of the amygdala in anxious individuals? Or is this something that if you look in all these other regions, you'll begin to see it as well? And is it more of a global type of thing? And what are the genetic underpinnings of this as well? So obviously there's a lot to be done. And I too am struggling with the question that you asked, which is, how do we really use this information in a way that's going to pragmatically impact our practice? But I am optimistic that that we can move in that direction, and I'm personally certain, obviously I'm invested in this, that the science needs to lead us there.
Okay, with that hopeful note, really wanted to thank you, Dr. Kalin, and also we're about out of time, so I'm going to turn it over to Dr. Lieberman for final words.
That was great, Ned. Really shows the way that your work sort of exemplifies the way the field has been trying to sort of build a neurobiological foundation to understand mental and behavioral phenomena and lead to better treatments. So it's very encouraging to see the progress that you've made and what the potential is for translating it into clinical practice in the future. And in these kind of otherwise turbulent times and somewhat ascientific attitudes,
Yes.
it's good to know. It's good to know that we basically have a strong empirical scientific foundation. So, thank you for doing this, and we hope to see you in person soon.
I can't wait to see you guys in person and be in New York. So, thank you again for having me. And if anybody has any further questions, feel free to contact me.
Okay, well, look, I think, the other relevant component, I mean, there's many touchstones, but the other, apart from the Intergenerational Center, is the Jay Gingrich's Center for Developmental Psychobiology, which is very much attuned to this. So a lot of potential interaction and interest. Thanks again, Ned, and be well, stay safe.
You too. Take care. Thank you.